Hi everyone, I'm going to present part of a project called Metagenomic exploration of mangrove soils disturbed with lignocellulose in plastic. I don't know how many of you have ever been in a mangrove forest, but they can look like this. These are salt-tolerant ecosystems in tropical coasts. They are mainly known for um, storing large amounts of carbon. They have a unique microbial diversity with specialized metabolic pathways that driving essential processes like nutrient cycling. Among the different anthropic pressures, mangrove forests are impacted or affected by plastic contamination. This is an escalating environmental issue because, or due to the durability and the resistance of these materials. And as a solution, it has been proposed that some plastics can be degraded or transformed by the microbial action, following similar pathways as with plant polymers like lignocellulose. Now, lignocellulose is the most abundant biomass on Earth. It is found in plant cell walls. It is a renewable and low-cost material with high impact or promising applications in the industry. It is composed of lignin, hemicellulose, and cellulose, forming complex structures that are also resistant to degradation. However, there are many microorganisms like fungi and bacteria that have some lignocellulosic degrading enzymes. These are cataloged in the Cazyme database, and these enzymes are also relevant because as evolutionary or through evolutionary mechanisms have the potential to also degrade other polymers like plastics. For example, um, this is a pathway found in an actinobacteria called Legionella sciensis that can use PET, a type of plastic, as primary source of carbon. Then mangrove forests, rich in lignocellulose because they are forests, and also contaminated with plastics, have these conditions that could support metabolic pathways for breaking both natural and synthetic polymers. So the objective of this project was to evaluate the effect of lignocellulose and plastics on the structure and functionality of microbial communities in mangrove soil microcosm. So we did experiments with microcosm, and for that we did a soil sample in a mangrove forest in Cartagena. There was a mix of all of those samples to have like a unified initial soil sample for the microcosms. Each experimental unit had 15 grams of that soil and 1.5 grams of an additional carbon source. It was rice husk for lignocellulose and PET for plastic, and a control without an additional carbon source. Also, for half of the samples, there was an addition of seawater from that mangrove forest, and we had a total of 6 treatments: lignocellulose with water and without water, plastic with water, without water, and control with water and without water. Each treatment had 4 replicates, and we had 2 sets of experiments. One incubated for 90 days and another one just for 30 days. Now, these are the treatments, and after the 30 and 90 days, the project had three phases. In the first one, there was a DNA extraction of all of the samples and all of the treatments to do an amplicon sequencing. In the second phase, there was a PET degrading consortia. That PET degrading consortia was built. And in the third one is the one that I'm going to talk about. But first, it's important to mention that after the first phase, the results show differences in the structure of the communities in the different treatments, and specifically there was an effect of lignocellulose and seawater. So we wanted to see or evaluate the functional potential of those communities, and specifically try to recover genomes and max to identify organisms with the potential of to degrade lignocellulose in plastics. For that, we did a DNA extraction of 3 replicates of the treatments incubated for 90 days. And this is the general workflow. We did a quality control, a co-assembly, a binning and a selection of marks, and then we did a taxonomic classification and functional annotation of those marks. Specifically, we use the KEGG database and the CASIME and PASIME databases, and also we did a taxonomic classification with the clean reads. So the results for the co-assembly and selection of marks, there's a lot of information, but I want to highlight three things. The first one is that we had on average 60 million reads per sample, from which we were able to co-assembly like the 30% of all of the reads. And from those contexts, we were able to recover 74 medium and high quality marks that represent 5% of all of the reads. This shows the complexity and diversity of the environment of the study. Now, the taxonomic classification, those are the results for the reads and marks. And again, I want to highlight three things. First, that in all the treatments without water, there was an enrichment of allotolerant organisms. After the 90 days of incubation, all of the treatments without water were dry. And this leads also to high salinity conditions. So based on our results, with those conditions and the addition of lignocellulose, we were able to enrich families like Micromonosphoraceae and Streptomycetaceae that are already known for being able to degrade complex lignocellulosic compounds. And in the MAX, we also see differences in the treatments with lignocellulose, but let's see the diversity evaluation. These are the results for the reads. We can see here a reduction of diversity, and specifically that treatment with lignocellulose and without water. These correspond to the enrichment of the two families that I showed you before. Here in the beta diversity analysis, we can also see that the treatments with lignocellulose are the most different ones, but specifically, again, the one with lignocellulose and without water. Now, here, the MAX, they represent only the 5% of all of the reads. However, we can see similar patterns in the beta diversity results. Now, let's see those MAX. To interpret the results, we group the MAX in 6 different categories based on their co-abundance patterns. And let's focus on the first 3 groups. This is for the MAX that are more abundant in the treatments with lignocellulose, either with water or without water. The group B are MAX that are more abundant in treatments with plastic control, but with water. And group C are the treatments more abundant, MAX more abundant in treatments with lignocellulose and without water. I am showing here 4 MAX that belong to this family and are going to be relevant in a moment. So the functional annotation, these are the results of a PCOA that is showing the clustering of the MAX based on the presence and absence of the 6,400 genes of the KEGG database. We can see here that most of the MAX share genes. However, we can see differences specifically in these 2 groups, groups A and B. But we want to focus on the pathways for lignocellulose and plastic degradation. So the annotation in the CASIME database, again, a lot of genes and a lot of family of genes, but to reduce the complexity, we did an overrepresentation analysis. We were able to identify some genes that were overrepresented in certain MAX, and also like half of the MAX were able to annotate at least, or to have overrepresented at least 1 gene in this database. Most of the genes overrepresented belonged to these 4 MAX were the ones that I was showing you. They are in the families of Cellulomonadaceae, Streptomycetaceae, Planktomycetaceae, and Lactobacterales. These are families already known for being able to degrade lignocellulosic compounds. And these are, or were grouped in the C group that are MAX more abundant in lignocellulose without water, and this one, lignocellulose with water. Now, the database for enzymes to degrade plastics, As we expected, not many coding sequences were able to be annotated in this database. Just a few MACs had one sequence annotated in this database. However, we were able to find two MACs with 26 and 25 sequences annotated here. And these are the MACs of the families Streptomycetacea and Cellulomonadacea. Again, these are the MACs that were most that had most overrepresented genes for lignocellulose degradation, were abundant under desiccation and high salinity conditions, and of course with lignocellulose. And this is really interesting because are microorganisms with potential for lignocellulose degradation, but also with potential for to transform plastics. So as conclusions, this study supports evidence that mangrove soils host vast and unexplored microbial diversity with potential for lignocellulose and plastic degradation. In this case, lignocellulose addition and the seawater were key factors in shaping the microbial communities. These factors act as selective pressures, favoring microorganisms with complex polymer degrading abilities. Genome-centric, which is basically a recording of MACs and genomes. This type of metagenomic analysis are useful strategies for studying complex environments like mangrove soils, and families such as Cellulomonadacea and Streptomycetacea show promise for lignocellulose and plastic degradation. This supports the idea or the evidence that some lignocellulosic enzymes might also degrade plastics. Okay, so thanks to all of the people that are collaborating in this project, and I don't know if you have any questions.
Thank you, Maria. Any questions from the audience? No questions? Maria, which sequencing technology did you use?
With Illumina.
Illumina.
Mm-hmm.
Yeah.
Okay.
Any other questions from the audience, guys?